Welcome to the United States Department of Energy Joint Genome Institute, JGI. The JGI was created in 1997 by the United States Department of Energy to unite the genomics and engineering expertise pioneered at 3 separate Department of Energy national laboratories. In 1999, the production genomics facility was opened here in Walnut Creek, California, in a partnership between the Department of Energy and the University of California. JGI's first assignment was to work on the Human Genome Project. The Human Genome was finally completed in 2003. JGI then refocused its efforts to use genomics to provide solutions in the areas of bioenergy, carbon cycling, and bioremediation. JGI is also actively engaged in education. Hi, my name is Miranda Harmon-Smith. Today we will show you how we sequence DNA here at the JGI's Production Genomics Facility. Let's start with a basic overview of how we sequence DNA. Our ultimate goal is to sequence all the DNA of a particular organism, or its genome. Let's imagine the cell nucleus as a train station. Inside our train station is the genome represented by long lines of train cars. The cars are the nucleotides, adenine, thymine, guanine, and cytosine. Our job is to make a map of which cars are in what order. The first thing we do is to break the long train into smaller pieces. This way, we can split the job up and work on sequencing all the smaller pieces at the same time, instead of trying to count millions of cars one at a time in order. It is faster to have more people working in parallel. There are 2 parts to sequencing DNA. The first is to identify each nucleotide. The second is to determine the order of each nucleotide in the train. We solve the identity problem by making special cars that fit the adenine, thymine, guanine, and cytosine positions, called ddNTPs. These ddNTPs fluoresce, or light up when excited by a laser. This way we can identify the nucleotides indirectly, without having to actually see the molecules. Imagine each ddNTP as a caboose that attaches to the end of a line of cars with a big colored lantern. Each fluorescent nucleotide replacement lights up with a different color, so that we can tell the identity of each. In order to tell the order of nucleotides, we make multiple copies of the line of cars. We start at one end and put a lit caboose in the place of the first car. Then we copy it again with a regular car in the first position, and a caboose in the second spot, and so on down the line. After a while, we end up with thousands of copies of portions of the train, each copy being 1 nucleotide longer than the previous copy. Because we only copy the train from one direction, the cars are all in the same order. They are just of different lengths. As you can see, if we line up the shortest train to the longest, we can read the last car of each train and see the identity and order of each nucleotide. That should give you a basic overview of what we do. Let's go into the lab and follow an actual DNA sample through the sequencing process.